Hello everyone. So today I am going to talk about classification of drugs. Drugs are classified in different ways depending on where and how the drugs is being used. So the method of interest to the medical chemist or chemical structure and pharmacological actions, which includes the site of action and target system. So let us see what is chemical structure classification. So drugs are grouped according to the structure of their carbon skeletons or chemical classification. For example, steroid, penicillin, and peptides. Okay, so they all contain the same carbon skeleton. Okay, but their pharmacological activities are different. So, for example, you can see steroid is a widely differing activities. Testosterone is a sex hormone, and spirolactam is a diuretic or fugistic acid in the antibacterial agent. So, classification by means chemical structure is useful for medicinal chemists, like who is a synthetic chemist or who synthesize the drug molecule in laboratory, but it is not very useful for doctors. So, doctors prefer the pharmacological classification of drugs. So, this classification is a list of drugs according to the nature of their pharmacodynamic behavior. For example, diuretics, hypnotics, respiratory stimulant, and vasodilator. So, this classification is particularly useful for doctors looking for an alternative drugs treatment for a patient. And third one is the World Health Organization has developed a classification based on the body system on which the drug acts, and that the classifier specifies 17 sites of drug actions. So the first one is the drug molecule which acting in the central nervous system. They call CNS drug. So the central nervous system consists of brain, spinal cord, and a drug acting on the CNS are psychotropic drug that affect mood and the neurological drugs required for physiological nervous disorder such as epilepsy and pain. Second is pharmacodynamic agents. These drugs that act on the body, interfering with the normal bodily functions. They include drugs such as vasculator, respiratory stimulator, and anti and allergy agent. Third is chemotherapeutic agent. Originally, these drugs were such as antibiotics and fungicide that destroyed the microorganism. Okay, and then finally, miscellaneous. These contain drugs that do not fit into those above three categories. They are categories as miscellaneous drug. So now let us talk about what is prodrug. So a prodrug is a molecule that is basically inactive. Okay, but when we it entered into our body and passed the blood-brain barrier, some chemical changes take place and it become an active drug. So let us see what is the definition. So products are compounds that are pharmacologically inert but converted by enzyme or chemical action to the active form of the drug at or near their target site. For example, L-DOPA or levodopa used to treat the Parkinson syndrome is the prodrug for the neurotransmitter dopamine. So dopamine is too polar to cross the blood-brain barrier, but there is a transport system for amino acids such as L-DOPA. And once the product entered the brain, it is decarboxylated. You can see here it is decarboxylating and then giving the active drug dopamine, which is the active drug. Now let us discuss about the stereochemistry and drug design. So we know the chiral molecule and stereochemistry has a wide application in the pharmaceutical chemistry. So it is well established that the shape of a molecule is normally one of the most important factors that affect the drug activity. Consequently, the overall shape of the structure of a molecule is an important consideration when designing an analog. For example, thalidomide. This is the classic case of enantiopure drug molecule developed in 1950s as a sleeping pill or originally marketed as a racemate mixture, not the enantiopure form. And that drug was found teratogenic, causing fatal abnormal replication. So let us see what happened. So this is the structure of the drug molecule, thalidomide. So you can see this is the S-thalidomide, which is a teratogenic agent, but the R-thalidomide is a potent sedative, or so it work as a sleeping pills. So this drug was marketed as a racemic mixture between 1957 to 1962 across 46 countries, and since the pregnant women consume this drug, So approximately 12,000 children were born with certain kind of disabilities. So you can see clearly in the photos that those children has certain kind of disabilities. So then now FDA came into the market and they told the drug molecule which is containing a chiral center, and it should be marketed only in enantiomer pure form, not in the racemic form. So this event is known as chiral switch. So we can see that ibuprofen initially it was marketed as racemic. Now it is marketed as the enantiomer pure form, the S form only, and we can see the improved activity of the drug molecule like faster onset and low adverse effect profile. Similarly, we can see in lot of other drugs, oxaprocin, ketoprofen, salbutamol or arbutamol, omeprazole. All these drug molecule initially was marketed in racemic mixture, but now it is marketed in enantiomer pure form. And we can see here what are the increased activity of the drug molecule or lower side effect of the other enantiomer. So why we need a single enantiomer of a drug molecule? So we can see that as I told now that R ibuprofen is inactive and S active ibuprofen is active painkiller. So since if we take the racemic mixture and it will the R ibuprofen will insert in our body, but it will not do any job. So why should we take extra chemical in our body? Similarly, R arbuterol and S arbuterol, R arbuterol is much better compared to the S arbuterol. So we should not take the S arbuterol and we should only take the R arbuterol. Similarly, R naproxen, S naproxen, and this dichloroprop. S dichlor this is a pesticide actually. So you can see the only R dichloroprop is effectively kills the weed, whereas S dichloroprop is inactive. So if we see the recent graph, okay, so this data is taken from Journal of Medicinal Chemistry 2024 published, and they have collected all the data from 2002 to 2022, and we can see that number of new molecule entities appear in the market, and we can see this red one is the racemic one or achiral one, and the blue one is the chiral molecule, and the white one is the racemic mixture. So if we see every year only blue like the chiral or enantiomer pure molecule is marketed. Racemic compound came very little amount in the market, one or two. Okay, so that is FDA actually approving the chiral compound only in enantiomer pure form to be marketed as a drug molecule. So why so? Like why one enantiomer is showing better activity or another showing inactivity or side effect? So in 1933, Asians and Seldom reasoned that differences in biological activity between enantiomer resulted from selective reactivity of one enantiomer with its receptor. So they postulated that such interaction require a minimum of 3 point fit to the receptor. So you can see that in the receptor they may have 3 point fit like A prime, B prime, C prime, and the drug with this particular binding group A, B, and C, only one enantiomer is fitting perfectly. But if you take the another enantiomer, you can see that A prime and D is a mismatch. So it is not point fitting perfectly. So it will not show the expected outcome or results of the drug molecule, or it may show side effect. So now, there are several examples of chiral drug molecules. Maybe we can categorize in intravenous anesthetic, local anesthetic, inhalational agent, neuromuscular blocking agent, some other drugs. So if we see examples of intravenous anesthetic, so you can see that etomidate, this drug molecule administered as a single enantiomer only, the R enantiomer, and site of action of this drug molecule is GABA receptor. So R isomer is 15 times more potent than the S enantiomer, and N enantiomer lack the hypnotic activity. This is the what is the structure of the drug molecule. So you can see this carbon is chiral, and particularly the R enantiomer is active. So why should we take the S enantiomer into your body, which will show some adverse effect? Similarly, ketamine. So this is the structure of ketamine, and we can see that S ketamine is 2.4 times more potent than the R ketamine as an anesthetic and analgesic agent. So R-ketamine, so emergence reaction with so if we take the R-ketamine into your body, so what it will do? It will do some side effect, and the side effect is hallucination, and vivid dreams and agitation. So metabolism of S-ketamine by liver microsome is 20% greater than the R-ketamine and 10% greater than the racemates, faster clearance of the drug molecule. So we can see here the S-ketamine is much more effective and useful for the treatment of the disease compared to the R-enantiomer. Similarly, this local anesthetic bupivacaine, so long-acting local anesthetic marketed as a recipe mixture initially. So because of that, a report of death is there. So bupivacaine introduced CNS toxicity and cardiotoxicity on accidental intravenous injection and difficult resuccination following cardiotoxicity. So you can see the one of the enantiomer is very toxic. So now L-dopabucin is marketed and R-upabucin is marketed. So this S enantiomer of bupabucin is a first pure enantiomer containing 99% of the S form. Now another drug is inhalation agent, so isoflurane. So some studies have found that the isoflurane. So this is the chemical structure. This is the chemical structure of the isoflurane. Okay, so isoflurane is 50% more potent than the R enantiomer, while other studies have found no significant differences. So both enantiomers are equally soluble in lipid bilayers. So S isoflurane induce 50% longer sleep time than the R isoflurane. Majority of the inhalation agent currently used as chiral except sevoflurane. Okay, so you can see, so mostly the inhalation agent are enantiopure in nature. So now let us know what is the role of enantiomer drug or chirality in the ADME process: absorption, distribution, metabolism, and excretion process. So absorption process. Let us see what is the role of enantiomer or chirality in this process. The stereoselective nature of some of the processes occurring in ADME may result in stereoisomer exhibiting different pharmacokinetic properties. In absorption, the rate of absorption of an individual pure enantiomer of drug that are absorbed by active transport may be different. For example, norgestrel, this is the structure, chemical structure of the minus norgestrel, is absorbed at twice the rate of plus enantiomer through oral and vaginal membrane, while L-DOPA is more rapidly absorbed than its D-L-DOPA. So you can see a particular enantiomer of a drug molecule has a much better absorption compared to the other enantiomer. Similarly, in distribution, so stereoselectivity appeared to have a very little influence though in the distribution, because distribution is happening through the blood, so there is no binding, proper binding or 3-point field model. So it just drug is getting soluble in the blood and it's just distributing. So that's why there is a very little impact of the stereochemistry in the distribution or transport. Okay. However, it has been shown that some enantiomer preferentially bind to a specific plasma protein in human. For example, R propranolone preferably binds to human albumin, while the S isomer prefer alpha acid glycoprotein. In contrast, stereochemistry may, depending on the mechanism of the transfer, influence the movement of drug across the membrane, separating one body compartment from another. Next, metabolism. So many metabolic processes are stereoselective, as we know that the enzymes or catalysts are uh, chiral in nature. So stereoisomers that are metabolized by this process may exhibit different behavior pattern. For example, the plasma half-life of S indocrinone is 2 to 5 hours, but the value for the R isomer is 10 to 12 hours. So we can see how the stereochemistry is influencing the overall metabolism of the particular drug molecules. Like one enantiomer is easy to metabolize, another is more difficult to metabolize. So these are other differences between stereoisomer are believed to be often due to the stereospecific nature of the action of the enzyme involved in the metabolic process. So this is the chemical structure of S endoconin and R endoconin. So excretion. So modern stereochemistry has been reported in the external. Basically, there is no much effect of the stereochemistry in the excretion process, because it is just again transporting through the kidney and just leaving the body. Now, solubility of drug molecule. So let us talk about the solubility of the drug molecule and how to improve the solubility, like water solubility of the drug molecule or lipid solubility of the drug molecule, and how we can increase that. Okay, so let us talk about that. So introducing conformal restriction. So let us. So now let us talk about the importance of drug water solubility in water. The solubility of a drug, both in water and lipid, is an important factor because absorption of drug from the GI tract, gastrointestinal tract, into the circulatory system by passive diffusion depends on them being water soluble. So, although the passage of the drug through other membrane will also depend on them having the correct balance of water and lipid solubilities. So a drug's distribution to the circulatory system and hence its action will also depend on to some extent on its reasonable water solubility. Once the drug has entered the circulatory system, either by absorption or by direct administration, its water solubility will influence its ease of transport to the body compartment available to that drug. So drugs that are sparingly soluble in water may be deposited on the root of their site of action, which can clog up the blood vessels and damage the organs, and it can cause the heart attack also. For example, many sulfonamides such as sulfomethoxazole tend to crystallize in the kidney, which may cause a serious kidney damage. In addition, to be effective, most drugs have to be administered in dose forms, and for that, the drugs should be very well water soluble. And since most drugs are administered in room temperature, and which is the body's temperature, so I think most of the drug molecule solubility should be measured in 25 degrees centigrade temperature or room temperature. So how to improve the water solubility of drug molecule? So let us discuss one more thing. So it is if the drug molecule is highly soluble in water, but it is not soluble in lipid or fat, this is also a problem because the drug molecule need to cross the cell membrane, and cell membrane has lipid also. So the drug molecule should be properly balanced. So in some circumstances, it should be noted that poor water solubility is a desirable property of a drug molecules. So let us know why. So although a reasonable degree of water solubility is normally regarded as an essential requirement for a potential drug, it's possible to use poor water solubility in drug action and therapy. For example, let us see that parental embonate, which is used to treat pinworm and hookworm in infection in the GI tract, it should be poorly soluble in water so that it can easily get absorbed in GI tract, okay, and retain in the gastrointestinal tract, which is the action site of the drug molecule. So how basically the drug actually react with water? So now let us show the structure of a compound will influence its water solubility and lipid solubility. So its water solubility will depend, okay, on what number of nature of the polar group, because we know the polar group number of more polar group is present when more polar the compound in water is. So in its structure as well as size and nature of the compound's carbon hydrogen skeleton. If a drug molecule containing a high carbon hydrogen skeleton like a heterocyclic group or aromatic group, it will be very poorly soluble in organic solvent. So in general, higher the ratio of polar group to the total number of carbon atom in the structure, the more soluble will be the compounds. So polar group that ionize in water will usually results in a higher water solubility than those that do not ionize. This we all know because water is there. Water will make H2 plus and OH minus. Okay, an aromatic compound would tend to be soluble in water than corresponding non-aromatic compounds. Okay, what is the factor that influence lipid solubility of a compound? So the lipid solubility of a compound depends on the nature and number of nonpolar group in its structure. In general, greater the number of nonpolar groups in the structure of a compound, the greater the compound lipid solubility. You know that is the organic molecule, hydrocarbon base, it will be more soluble in lipid. Consequently, the lipid solubility of analogs may be improved by replacing polar group by significantly less polar group to a non-polar group. Okay, so potential drug candidates whose structure contain both polar and non-polar group will be solubility in liquid and water. So one of the most important thing, how we can improve the water solubility of the drug molecule. Let us discuss. There are several ways that we can improve the water and water solubility of a particular drug molecule, keeping its activity same, or we can even increase the activity because since it is more water soluble, it could be easily absorbed in our body, distribute in our body, and metabolize in our body, and excretion also easy. So let us see what are the possible ways. So first one is salt formation. The salt formation usually improves the water solubility of acidic and basic drug because the salt of this drug dissociate in water to produce the hydrated ions. Hydrogen and hydroxide ions can disturb this equilibrium if they combine with the appropriate cation and anion to form less soluble acids or bases. Consequently, the pH of the biological fluid may affect the solubility of a drug and as a result, its activity. Okay. So in general, increasing the hydrophilic nature of the salt should increase its water solubility. So in salt formation, the activity of the drug is normally unchanged, although its potency may be different. So you can see that if we make a salt of particular drug molecules, its activity will remain the unchanged. Okay, although its potency may be different, maybe decrease or increase, but most of the cases it is increased because it will easily absorb and distributed throughout the body. Second one is incorporating water stabilizing solubilizing group into the structure. So the incorporation of a polar group into the structure of a compound will normally results in the formation of analog with a better water solubility than its parent lead compound. For example, the incorporation of strongly polar alcohol like which are all readily water soluble, amine, amide, carboxylic acid, sulfonic acid, phosphorus oxy acids group which form relatively stable hydrates with water would be expected to result in analog that are more water soluble than those formed by the introduction of less polar ether, aldehyde and ketonic functional group. And then the third one is the use of a special dose. So if we can bind the drug molecule with some other additives which are highly water soluble, then that will helps the drug molecule to be more soluble in water. For example, we can use a co-solvent, a colloidal particle, a surfactant, a myces, liposome, okay, or complexing with cyclodextrin. So if we bind or encapsulate the drug molecule in this kind of more water soluble compound, then the solubility of the overall solubility of the drug molecule will increase. As I told, introduction of a residue containing carboxylic group is one of the easiest way to improve the water solubility. So we have a drug molecule called sulfathiazole. It is used as an antibacterial drug molecule. So this drug molecule is not very much water soluble. So we can easily make it is more water soluble by incorporating a carboxylic acid group. So how we can do that? So if we react this drug molecule with succinic anhydride in ethanol, so you can see this NH2 can attached with this carboxylic acid, and it becomes succinyl sulfathiazole, which is also working as an antibacterial agent, but its potency will be increased because now it is more water soluble, which will be more easily absorbed, distributed, okay, and metabolized. Another example how to improve the water solubility incorporating a carboxylic group is Dapsone. This is also an active antibacterial leprocytic. Okay, so if we react this kind of molecule with CH2 carboxylic acid, and we can see we can after incorporation of the drug molecule we can improve the water solubility, but the activity of the drug molecule definitely will increase. Phosphate group. So the number of phosphoric acid halide derivatives have been successfully used to attach phosphate group to hydroxyl group of a particular drug molecule. So if our drug molecule have a OH group, that could be aromatic OH group, that could be aliphatic OH group, we can simply attach to a phosphoric ester. So these three are the most commonly used phosphoric ester, which we can attach with the OH group. And the next step, we can just simply hydrolyze the ester to get the free phosphoric acid group, and this free phosphoric acid containing drug molecule is way more easily soluble in water compared to the free drug molecule. Okay. So this is how it is possible to increase the overall water solubility of the drug molecule by incorporating a phosphoric acid group using this kind of phosphorate ester. Okay. The third one is another functional group we can incorporate in the parent compound to increase its water solubility. That is sulfonic acid group, SO3H group. And the easiest way to react the parent molecule with sulfuric acid. So sulfonic acid group can be incorporated into the structure of lead compounds by direct sulfonation with concentrated sulfuric acid. You can, for example, 8-hydroxyquinoline, if we treat with sulfuric acid, we can simply incorporate SO3H, and then after various steps, we can simply incorporate the iodine, which is essential for the drug molecule. And we can see this 8-hydroxy 7-iodo 5 quinoline sulfonic acid is a potent topical antiseptic. Okay. So alternative route are addition of sodium bisulfite to the conjugate double bond, and the reaction of primary and secondary amines with sodium bisulfate and methanol. So let us see how we can improve. So if we see this kind of drug molecule we have, and we have a unsaturated double bond here. So if we react those drug molecule with sodium thiosulfate, we can simply make it SO3Na plus functional as drug molecule, and definitely this drug molecule will be much more water soluble compared to the parent molecule, and it is working as an antibacterial agent. And similarly, if we have a drug molecule containing NH group, if we react that one with sodium thiosulfate in formaldehyde, so we can see after formalization to the NH position, and then after that SO3Na group is attached, which is much is a salt, so it's a much way more water soluble compared to the parent molecule, and it is usually used as analgesic antipyretic drug molecule. Incorporation of basic group, that is also another way to improve the water solubility of the drug molecule. So water solubilizing group containing basic group can be incorporated into a lead structure by the alkylation and acylation of alcohol, phenol, and amines. So alkylation is achieved by use of alkyl halide whose structure contain a basic group, while the acylation usually involves the use of acyl acid halide or anhydrides. So in both alkylation and acylation, the basic group in the precursor structure must be either unreactive or protected by suitable groups. Okay. And amide derivatives are usually more stable than the ester. That is, we know that ester can often rapidly hydrolyze in serum. The reaction between the catalyzed by serum esterase. Okay, so in these cases, the analog effectively act as a product. Okay, the introduction of water solubilizing amino acid residue using standard peptide chemistry preparation method has also been used successfully to introduce basic residue. So you can see the Mannich reaction is frequently used to introduce basic group into both aromatic and non-aromatic structure. So you can see there's an example. So you can see we have this PHCOCH2CH3. If we just do the formylation reaction using diethylamine, and we can just make this kind of basic incorporate basic group. Then after various steps, if we incorporate this CH2O COCH3 group, you can simply make it more water soluble, which is used as a narcotic analgesic. So now the incorporation of water solubilizing group in the structure. Let us see more example. So here we can use that we can using N alkylation of this drug molecule on azaphenothiazine using sodium hydride, so not sodium hydride, sodium amide, and then we can see that we can simply incorporate this group, and it will definitely increases water solubility. Similarly, we can do the O alkylation of the reaction, and we can see after alkylation, we can get the functionalized OH group here, and which is definitely more water soluble drug molecule, and O acylation. So if you have this kind of OH group containing drug molecule, is like the metronidazole. If we react this with this acid chloride containing group, it will make an SN2 attack here, and the ACL will go out, and we can get this functional group containing drug molecule, which is used as a anti-protozoal, and it is much more water soluble compared to the parent molecule. So in acylation, so we can see starting from nitrobenzyl chloride. If we treat with this long chain amine, we can simply incorporate this long chain amine here, and then if we reduce this nitro group to an H2 group, we can get the procainamide. This is used as a local anesthesia. So this is how actually we can incorporating different alkyl group or acyl group in a particular functional group, we can increase the overall activity of the drug molecule by more making it more soluble in water. So let us see how we can increase the water solubility of this particular drug molecule. So you can see that if we take 2-methyl phenol, and if we treat this one with NaOH, we'll make a O minus Na plus sol. Then if we take this long chain OH containing group, you can see that polyhydroxy group is there here on hydroxy, here on hydroxy. And if we treat this one with here, so Cl will replace, and we'll get this polyhydroxy containing drug molecule, which name is menfeshin. This is a muscular muscle reluctant. Okay, so similarly, this thiophenyl, this is a bronchodilator, okay, and this drug molecule simply we can increase the water solubility by incorporating OH group. So how we can incorporate the OH group? So if we react this drug molecule with a epoxide, so you can see this NH is nucleophilic and this is electrophilic center. So it will attack this one and this electrophilic center, and it will open the ring, and it will attached with the OH functional group, and which is very important drug molecule for bronchodilator, and the name is etoflurane. Okay, similarly, we can react this to parent drug molecule with different other epoxide to incorporate various functional polyhydroxy group here, and the name of the drug molecule will be proxyphenylene, the bronchodilator, and diphenylene bronchodilator, like that. Okay, so this is how we can use epoxide to incorporate the OH group in the parent drug molecule to improve its water solubility and activity. Similarly, if we have guanidine, you can see if we incorporate a OH group here, functional group. So first, how we can do, if we use this reagent here, and then then finally after adding this one in the nitrogen, if we clip this one, we'll get free OH group, and which is a very good drug molecule as used as an antiviral drug molecule. So other way is we all know that sugar molecule is highly water soluble. Okay, so if we can attach to the drug molecule with a sugar molecule, which will not have any side effect in our body, or it will not reduce the overall activity of the drug molecule. So that way we can simply increase the water solubility of the drug molecule. So if you have a drug molecule like that containing NH2 and SO2 and H2 group, and if we you attach this drug molecule with the glucosamine, okay, so we will get this glucosyl sulfilamide drug molecule, which is which is used as an antibacterial drug molecule. Similarly, if we have this anti-tuberculosis drug molecule, and if we attach to D-glucuronolactone, and we can see by attaching this drug molecule, sugar molecule, you can overall improve the water solubility of the drug molecule. Here, this drug molecule simply we can attach to it the D-glucosamine, and we can improve the overall water solubility of the drug molecule. Okay, but it will not have any side effect, but it will have positive effect on it. So the effect of pH on the solubility of acidic and basic group. So the acidic or basic pH will either enhance or reduce the ionization of this drug with subsequent changes in their solubility and absorption through membrane. So the degree of ionization of weak monobasic as drug, monobasic acidic drug at different pH values mainly calculated using the Henderson-Hasselbalch equation. So we all know that basic BSc chemistry only. So how we can calculate the pK value? pK is equal to pH plus log concentration of non-ionized form divided by concentration of ionized form. So this is where we can calculate the pKa value of a drug. So now let us talk about the surfactant and amplifiers. So amplifiers are molecules that contain both hydrophilic, which is water-loving group, and hydrophobic, which is water-fearing parts. So this dual nature allow them to interact with both water and oil phase, making them essential component in various biological and industrial processes. Now let us talk about what is surfactant. Surfactant are compounds that lower the surface tension of water. Their structures contain both strong hydrophilic polar group and strong hydrophobic non-polar group, and so they dissolve in both polar and non-polar solvent. So in drug molecule, there are several surfactant has been used. So you can see this kind of cationic surfactant, long chain. Okay, carboxylic acid, salt, sodium stearate. Okay, so you can see anionic surfactant. So if the drug mole surfactant contain NH2 group, simply treating with HCl, we can get this kind of anionic surfactant. Okay, so these are the several examples. Okay, how to develop surfactant and which are used in the drug molecule. So in biological system, surfactant dissolve in both the aqueous medium and the lipid membrane, and so tend to accumulate at the interfaces between those phases. This property is the reason for the antiseptic and disinfectant action of some non-ionic and quaternary ammonium surfactants. So surfactants such as cetylpyridinium chloride and benzalkonium chloride, okay, partially dissolved in the lipid membrane of the target cell. This lowers the surface tension of the cell membrane, which results in lysis and the death of the cell. So let us give some examples how the disinfectant work actually, or the antiseptic work. So you can see that octohexanol 9, so this is the octohexanol 9 here, okay, and here, okay, and the no, non-oxidil 9 are different form of the surfactant antiseptic. They do not dissolve in the cell membrane of pathogen. They dissolve in the cell membrane of spermatozoa. This immobilizes the sperm, which allow this compound to be used as the spermicide in birth control. Okay, so if you see, if we have this kind of spermaticide, and if we just make a long chain here, incorporate a long chain here, and then if we make a surfactant out of that, you can simply act as a spermaticides. Okay, same here also. So these are the some other example how introducing a long chain here. This is the polar head group here we can see, and this is the non-polar head group here, tail group here. So this is how we can simply develop a surfactant which will be easily soluble in water as well as in lipid, showing its better activity as a drug molecule. So now let us talk about the micelles. Okay. So as the concentration of a surfactant dissolved in water increases, the system changes from the true solution to a colloidal solution. The surfactant molecule find it's more energetically favorable to form colloidal aggregate known as micelles, in which the hydrophobic section of the molecule effectively form stearate organic phase of the hydrophilic part of the molecule in the aqueous medium. So this is the graphical representation how the missiles are formed. So you can see the hydrophobic end, this one, and this is hydrophilic end. So you can see how it is from the spherical missiles. Okay, increasing, if we increase the concentration, it will form cylindrical missiles. Okay. Okay, and then finally, if we still increase the concentration, it will form laminar missiles. Okay, these are the different shape of the missiles, depending on the concentration we can form. This is not that important. Okay. So thank you very much for attending the class.